I have the pleasure to introduce Dr. Jan Eric Lee. Dr. Lee joined Washington University School of Medicine in 2023 as an assistant professor in the Department of Nerve Surgery and Genetics. Dr. Lee is a member of the Brain Tumor Center, Pipeline Cancer Center, and Open Center for Neurological. Dr. Lee is PhD in computational biology and bioinformatics at King College University School of Life Sciences under the supervision of Dr. Lee Jones lab and completed his postdoc at Dr. Dean Wren's lab of gene regulation at the Ludwig Institute for Cancer Research at the University of California, San Diego. His research interests focus on developing innovative computational tools, methods, and utilizing single-cell epigenomics to gain an understanding of gene regulation in mammalian models and human disease. Especially brain disorders like genetic brain disorders, glioblastoma, drug addiction, and Alzheimer's disease. As a participant in the BRAIN initiative, Dr. Lee has led efforts to map epigenetics landscapes across thousands of distinct cell types in human and mouse brains. Dr. Lee has developed computational tools for integrating single-cell multi-model data sets at a million cell scale. In July 2024, he received the St. Baldrick Scholar supported by the Joe Strong 71 Journal Fund. Today, he will be presenting on single-cell epigenomic analysis of mammalian burden and neurological disorders. Thank you, Dr. Lee, for joining us. And with that, I will hand it to him.
Right. Okay, I want to thank everybody can hear me. I want to thank Mona once again for inviting me. This is such a nice trip here. And I also want to big shout out to Tamara and Nathan together events. And also thanking for the students, the faculty member meeting. This is such a nice community of a lot of fun as well. So thank you once again for coming. So today I'm gonna share a little bit about my previous work, but also have something ongoing works in my lab. I'm in general interested in brain neurological disorders. I'm pretty sure I can have a lot of shared interest here. Thinking in the future probably will have more collaboration. So today the topic is single cell epigenomic analysis of mammalian brains and neurological disorders. Okay, I want to start it from one quick question. So, what does the brain look like? I would like to know my answers. I think the brain is actually like a building, like here, and then I would like to know why. So, in my opinion, well, brain are built over time, right, from bottom up. For sure, a building also built like that. And then another very important reason is brain architecture is actually compressed or billions of connections between neurons or across different areas of the brain. If you think about a building, it's a similar thing. There's tubes, there are electronic wire, they're all connected to each other between different rooms. So if you take this concept and then put it into a, well, there's a model, you might open an eye, it'll give you a little image like that. So you'll see the, this is like a building sector, a structure in a brain shape, right? They have different rooms, they probably have different functions. Actually, the brain exactly function like that. There are certain brain regions responsible for sensing the environment, or some brain region there are making decisions. So in a room, how can we tell this is in function? So we simply turn on light. For example, here we turn on light, so we know somebody is using this for giving talk. This is also true for gene regulation, but it's not that simple. You just press that surface, right? There's a tons of elements, modifications, they're all getting involved to turn the genes on or off. So we're really interested in study epigenome.
Excuse me. Can you have the microphone more, like, can you increase the volume of the microphone?
Point it up higher.
Oh, point it up higher.
Yeah, they can't hear in Zoom.
It seems that the microphone is pointing to your chest. You can get the cone of it to point further.
Is this better?
Oh, that one. Oh, the other one. Okay.
Really encourage everybody to move.
If it's uncomfortable, we want it to be uncomfortable, just like.
No, but
No, it's okay.
I use the other mic. Yeah,
Um,
you can use the mic. You can use the boom mic, it's just there. If it's in
One that is on the stand, just.
Oh. the podium.
Oh,
No, it's-
If you want to use this, like, we can turn this on. And is that-
Yeah, then you don't have to
Yes,
hold it.
not
Yeah,
the-
yeah. Okay.
it's better?
Yes,
Okay, you don't sound good.
thank you.
I'll turn this off first. Yeah, so the gene regulation is also a lot of players get involved, right? Many modifications, transcription factors all get involved, so that's why we're interested in epigenome. So sometimes either the light or the switches could be broken, so this room probably need a fix, right? This is also true in gene regulation. If something behavior is weird, well, it probably cause a lot of disorders, including brain tumors, schizophrenia, depression, Alzheimer's disease, bipolar. I'm sure there's many other neuropsychiatric disorders are also got affected by gene dysregulation. So the good news is, let me show you several numbers. So first of all, I'm trying to do a little research in the brain tumor, and then I searched several numbers. It's actually very scary to me. Actually, there is more than like 80,000 of individual diagnosis as a certain type of brain tumor in the United States. And then usually it costs a lot of money to treat it monthly. And then even you have a drug, even you have a treatment that works for a certain cancer type, it usually doesn't work for brain tumor because we have this brain-blood barrier, and then the delivery of the drug is the main issue. And then if you take a look at the estimate by WHO in 2022, they said, they published on 2022, they probably finished the survey before that. It says, well, 1 in every 8 people in the whole world, they probably will suffer one kind of mental disorder in their life. I don't really have it, I'm fine, but who knows in the future? I mean, the real issue is you have to suffer that for 15 or 20 years. You have to take the pills. So this is truly a top burden right now in the United States and also in the whole world. So the good news is people are interested to understand what is the genetic basis of those mental disorders. And then there's a lot of genome-wide association studies, right? They're trying to sequence different cohort, they're trying to identify the risk variants which associate with certain trait or neurological disorders. Well, for some of the risk variants, it's very hard to interpret if they're located in the coding region, there are missense mutation or nonsense mutation. It is easier to figure out the reason. But actually, many of them, actually larger than 90% of those risk variants are not located in coding region, they're in non-coding regions. And then they're supposed to play a role in the gene regulation. That is the real challenging part. I think people focus on the coding region for another reason because in the early days, it's just the whole genome sequencing is very, very expensive, right? You just don't have the chance to look at those non-coding regions. Well, there's an estimate from this Nature paper in 2021. Actually, among those 90% of the risk variants in non-coding region, probably 50% of them, they're located in the cis-regulatory element and then affect the gene regulation. So I draw a little paradigm here, just trying to give you a basic idea about in my opinion, the main questions in the field. So first of all, we really want to identify where are those switches in the genome, or we call it cis-regulatory elements in the genome. I guess that's part of the reason we do a lot of sequencing. We do a lot of chipset sequencing, attack sequencing, RNA sequencing. We try to figure out where are those elements and then what is the genome responsible to that, right? And then the second major question, in my opinion, it is because eventually we care about the genes, care about the proteins. We really want to figure out what is the potential target gene for those CREs. And then the last question, like I said, many of those risk variants actually located in the CREs where there's a broken in those switches in genome. So they affect the binding of the transcription factor or the chromatin regulators. That's why you see the gene tag regulations. So we really want to figure out how those risk variants affect the gene tag regulation so that you can think about a way to correct that. For example, using CRISPR-based method. The good news is there are already tons of sequencing technologies help you to profile different signatures from the genome, including chipset, attack, DNAs, whole genome bisulfite, Hi-C, they all capture different signals and have been well adopted by many consortiums. Right? But the real, the major issue is most of these assays is developed to profile the bulk level signals to carry that. For a certain tissue have a lot of heterogeneities, you are counting on the single cell technology to further dissect the gene regulations. I guess I don't need to put too many words here, but I do want to pay your attention. Well, nowadays it's not only the RNA-seq, single cell RNA-seq or single cell attack-seq. Actually, we have spent the last couple of years having developed new technologies. You can profile different signals from the genome, either alone or coupled with the RNA expression from the same cell. So for example, I'm contributing to the first version of the pair tag, we can co-profile the histone modifications as well as the gene expression from the same cell. I collaborate with Joe Ecker's group, they can profile the DNA methylation from the single cell or even have the Hi-C signals as well. They name it as single cell methyl-3-C-seq. So thanks to this single cell multiomic tools, we can really look into the tissue heterogeneities. I will show you one example here. So this is the study. It's truly a team effort. We request the tissue, and then we share the tissue in different institutes so that we can make the data very consistent. But basically, what we did is we dissect the human brain, and then we get 42 brain regions. They are coming from 3 neurotypic adult donors. In total, we are able to capture the open chromatin profiles using SNAP attack developed by my friend Sebastian. We are able to capture the open chromatin profile for 1.1 million human brain cells. So once we collect this data, right, the first task it is to do the cell cluster.
Actually, the donors are the natural dead donors from the old people. Do you have any idea for the donors?
Yeah, for those 3 neurotypic data, they're all normal conditions, and then they're not the old people. It is a very, we set a very stringent cutoff, so what is the normal? And then usually the people suicide themselves not considered as normal because there could be potential mental disorders during their life. And then, yeah, and then even you have all of this screening, then if you check the morphology of the brain itself, maybe it's not very well, certain areas bigger or larger is not very well aligned with the reference. We also don't consider it as the normal brains, yes. And then just unfortunately, we are not able to collect the data from both sacks. Those tissues are only came from 3 miles. We waited for a couple of years. We tried to also collect the female samples, but I mean, the tissue bank just say what I can do, like the young female just doesn't die. Yeah, they're smarter than us, but that's the reason we're not able to. Thanks. So like I said, the first task it is to do single cell clustering, right? For the people familiar with the single cell RNA-seq analysis, the idea is quite similar, but for the TAC-seq data, it's slightly different. So for example, you can imagine there's a matrix, right? So first of all, we're dealing with millions of cells. We're not dealing with hundreds of cells. So this is a huge matrix. And then the second reason is if you are interested in RNA-seq, we probably care about 20K-ish protein coding genes. But now at TAC-seq, we actually care the signals across the whole genome. We're not only care about certain genes. And then because we're capturing the signal from the DNA itself, right? The DNA in normal cells, you only have 2 copies, right? So it's very, very rare you capture 2 copies, one in one cell and then in a certain genomic regions. So it's very likely you just capture 1 read for a certain region in a single cell. So you can imagine this is a huge matrix. At the same time, it's also a binary matrix. So they make the analysis very challenging, actually. To load this matrix, it is challenging. Most of the algorithm will have a memory issue, and then even the algorithm works, it will take a long, long time to process the data. So for the last couple of years, I actually worked with Rongqing. We developed the first version of the computational tool called SnapAttack, and then Kai is taking over developing the second version of the SnapAttack. I think the idea is we first cut the genome into 5 KB beans, and then we count the reads for every bean, and then we're using this cell by bean matrix for downstream analysis. But still, it is challenging to handle this huge sparse matrix at the beginning. So we first split this matrix into half. One is we random sample 300K cells, save them as a landmark cell matrix. And then the rest of the cells will save as a Curie cell matrix. And then we only do the dimension reduction. For example, the PCA, for example, the diffusion map. We do the dimension reduction on the landmark cell matrix. And then how to find the location for the cells in Curie cell matrix in the low dimensional embedding. We actually later find an algorithm called a Neurological algorithm. This is a very efficient algorithm. You can project the cells from the Curie cell matrix to the low dimensional space. So in this way, we're able to have a measurement of the similarities between every cell. So we do the downstream clusters starting from there. Another thing I really want to mention is actually when you are working with single cell data, there's a parameter actually is very, very important if you're using graph-based clustering method, for example, Latent algorithm. So there's a parameter called resolution. If you change that parameter from 0.1 all the way to 1, the algorithm will give you a different number of cell clusters. But which one is the right cutoff or the right resolutions? I think this is very crucial for downstream analysis. So I took a very straightforward approach. So for every resolution, we just do thousands of time of iterative run with a different starting seed. So in this way, you can generate a consensus matrix. So here I'm showing you a little matrix at every resolution. Each row and each column is a cell. If you see the signal is close to dark red, that means across thousands of times of iteration, cell A and cell B, they always cluster together. And then if you see a dark blue, that means cell A and cell C will never cluster with each other, right? So you can tell from the resolution 0.1 all the way to 0.5, you can see very robust clustering effects, right? But if you keep increasing the resolution to 0.6, you are starting to see some of the cells doesn't know really, like, actually belongs to this cluster or the other cluster, right? I will do the clustering more conservative way. So in this case, you can calculate some measurement from this consensus matrix so that you can find a local maximal or local minimal to find a more reasonable cutoff. So in this example, we just picked the 0.5 resolution as the cutoff. So long story short, we do this rigorous analysis on the cell clusterings. We're able to solve 100-ish cell types. They are grouped into three major clade: glutamatergic neuron, GABAergic neuron, and non-neurons. So once we get the cell cluster, one most important thing is to annotate cell energy of those clusters. So we're counting on the signals, not only on the promoter, but also on the gene body. I will show you one example. So here, I name a cell cluster CHO, but basically it represents cholinergic neurons. We're actually checking the open prompting signal at the gene locus CAT. This is a well-known marker for this gene, for this cell type. You can see this gene actually have no signal in the other cell types. And since we know where are those cells coming from, which is the exactly brain region we collect those cells, you can also check the brain region information to further confirm this is the right cell type from right brain regions. For sure, we also integrated with the single-cell RNA-seq data collected by Allen Institute. They are generating a huge cell atlas using RNA-seq. We also do the integration to further confirm the annotations.
I have a question.
Yeah.
So in this, the glutamatergic neurons in the cortex have both the vehicle G1 and the vehicle G2. But the cortical neurons is not to express only vehicle T1, not the vehicle T2.
Yes, very good. Yes, it's not only coming from the cortical regions, I think.
But the neurons you labeled as intraepithelial neurons, these are cortical neurons.
Yeah, those are only came from cortical regions.
They should only have vehicle T1.
Yes.
Not the vehicle T2.
No.
But if you have both.
Yeah, we are not only dissecting the cortical region, we also have the
It's both, it's only the cortical.
Oh, you mean the upper clade? Yes,
that-
the upper clade only coming from the
So
cortical
there's
regions.
two column, glutamatergic neurons. So you are showing both vehicle T1 and a vehicle T2. To me, it's like anatomically, I don't understand.
I think we definitely captured both big root 1, big root 2 neurons.
But in the cortex, it's only vehicle T1.
Yes, I understand that. So this is the cluster based on the open prompting signals. It's not, I will tell you like if you do the cell clustering based on different modalities, the clustering modality is slightly different. For example, Wickle 2-1, Wickle 2 could be an example, but for the D1, D2 MSN cells, they also cluster very differently if you're using RNA-seq and tag-seq. So if you're using RNA-seq, the D1, D2 separate very well between each other. And then you will see a gradient difference across the different nuclei in the cerebral nucleus. But if you're using a tag-seq, the D1, D2, they always cluster together. I think they probably both true is the definition of the cell types. So
No, only one can be true. Only one can be correct. Like cortical neurons. Cortical glutamatergic neurons express only vehicle G1, not vehicle G2.
Yeah, but that is only like one or two features or marker genes we're looking into. Well, we can
Is like detecting both. So it cannot be like, I just don't understand.
There is a possibility that the chromatin is open, but gene expression is not there. So the ATAC is only detecting the chromatin openness, but it doesn't mean that the gene is expressed. Gene expressed.
Oh, that is the question. Okay. Oh, I can explain that. I'm sorry, I'm misleading your question.
That's it,
So,
that's it. Yeah. Okay, okay.
yeah, I think you're right. Well, thank you for that. Yes, I think I should make that clear. So if you see the signal here at the promoter, it doesn't mean this gene is expressing like in the certain cell population. Because the tactic just measuring the open chromatin region, this DNA is exposed.
But if they're not expressed, why do they have open access?
Yeah, to initiate the gene in transcription, you must have an open promoter at the first place, so that the TF can bind there and PO2 can bind there to start the RNA transcription.
But then, like in vehicle G2, they are not expressed, so they should not have access.
It's possible that requester might be bound to locate.
Yeah, the opening of the chromatin is one thing you need to start the RNA transcription. You also probably need certain chromatin contacts. For example, you have a more active state in that chromatin region to start the transcription. I think overall, tactic is not a good measurement for gene expression, but they do offer different informations. For example, many of those potential cis-regular elements in the non-coding regions. Okay. Yeah, I can explore more later. Yes. So we just so the next step we try to call the peaks from those open chromatin profiles, and then we end up with roughly 500,000 of the candidate cis-regulatory elements from this data set. We did a brief summary of those. We actually try to do the genomic annotation for those CREs. Actually, only I would say 10% of them located at the promoter. It is the region I just showed you the signal as a promoter. But actually, there's many of those open chromatin region. They are located in intron or intergenic region. They're considered as the non-coding regions. Actually, I want to point your attention to the annotation for many transposable elements there. To our surprise, actually, there are 20-ish like CREs are located at the genomic region have been annotated as line, sign, and LTR regions. There are transposable elements in the genome. This is very interesting because I think there's a report earlier said the transposable elements contribute to the brain development in the early state. So we are also curious about that.
Just offhand, how does this relate to the just random distribution, the background distribution of these elements in the genome? Like which ones are overrepresented?
Which one is represented? So
like what fraction of the genome it's intron, energetic, or line of sign? What's the relative like enrichment I'm trying to say?
We actually calculate the genomic annotation across the whole genome region. We have
Are you asking what the null?
Yeah, the null, yeah.
What's
yeah.
the null?
What's the null?
no.
The null.
Like if it was even
if
uniformly
it turned out that exactly 27.8% were intergenic, you'd say, oh, you got a null distribution.
Yeah, yeah.
Oh, this is the percentage from all the CREs we identified.
Right, but if okay, what are the annotation? What's the distribution of the annotations over the genome? What's the background distribution of annotations relative to which ones are enriched? That's what.
I see. I would say the coding region probably 5%, and then the 90% will be the non-coding region, including those transposable elements. That's the non-distribution.
let's say, could be a random. For example, promoter is 5%, but we know that only 3% are the gene, and only certain percent of this 3% are the promoter. So there's actually enrichment in the promoter. But you
Oh.
want 5%.
This is not
promoter
an
is, let's
enrichment.
say, less than maybe 0.1% in the
Yeah,
entire.
it's not an enrichment, yeah.
It's not an
You're
enrichment.
gonna show an enrichment, then I didn't mean to derail you.
Yeah, it's not an enrichment, just show the proportion of the CREs.
So what's the size of the promoter in the TSS region?
For human, we define it as 2 kb upstream, 2 kb downstream.
Then definitely TSS promoter and the TSS is highly enriched, right? In terms of this.
It's not expected.
Yeah, but here I'm not trying to show the enrichment, just show the global proportion of different elements. I think we will also compare with the brain data collected by ENCODE. I think this is the power of single cell data set, where we actually identify more that's expected. Okay, there are 47-ish CREs you never see in the bulk level taxid. I can give you several examples. Most of those are found in progenitor cells, from subventricular zone, subgranular zone, something like that. You'll find novel peaks there. So the next thing, we want to cluster those CREs into different groups. So I show you a big heat map. Every row is a major cell population. Every column is a CRE identified. So I did this unsupervised clustering. You see most of the CREs are quite cell type specific. Only a small proportion, maybe 5% of them, are consistently accessible across all the cell types. And then most of those CREs are located at the promoter regions. I guess that explains why you see the weak group 1, weak group 2, sorry, extending your question. So this evidence basically show, well, the CREs are more cell type specific than the genes. Actually, not only you can see the difference between different cell types, since we collect the data from different brain region, we can also explore the regional specificity of those CREs. I show you one example is the oligodendrocyte. We did a cluster, we clustered them into 3 major subpopulations, including mountain-forming oligodendrocyte, and return oligodendrocyte. So if you color the cell based on the brain region we collected, including cortex, cerebral nucleus, pons, cerebellum, hippocampus, thalamus, and midbrain, you see there's a slightly gradient difference in this cluster. So at the beginning, we don't trust it because people usually consider glial cells are quite consistent across the whole brain. We're probably thinking this is maybe a battery effect or like a donor effect. But actually we checked, the data we collected from different donors, they're quite well aligned with each other. So maybe this is biology. Maybe there is a difference for those glial cells in different brain regions.
So middle brain, you just you lump it together as a middle brain.
We only dissected, yes, we are able to dissect more.
Middle brain has many subregions, has very different cell types.
Yes, I think this is the study in the first phase of the brain initiative. This is the limited sample we can collect it. In the second phase, they are trying to dissect every subnucleus from the brain. Yeah, they're trying to do more detailed annotations. For the mice, we already have that little dissection. So if you do the differential analysis for those CREs across different regions, and then I apply the k-means cluster, you can see actually there are thousands of peaks are quite specific in certain brain regions. And the signal is quite consistent across 3 donors. And you see some of the, not only see some of the peaks are consistently accessible in certain brain regions, you also see some of the signals are lost in certain brain regions. This is to our surprise. We actually, the glial cells is not that similar across different brain regions. I think the data from both donors prove that. And this is The regional specificity for glial cells is not only true for oligodendrocyte, we saw similar patterns for astrocyte, for oligodendrocyte precursor cell, for microglia. We actually think this is very interesting because many of those, for example, brain tumor, they are derived from glial-like cells. And then if the brain tumor are growing in certain brain region, they could be very visible. For certain region, could be fine. So this is probably explains why different region, the brain tumor coming from different region have different phenotypes.
Do you see the same pattern for epithelial cells?
Epithelial cells, we're not able to capture that for this study.
That would be a very good negative control.
Yeah, so in the mouse data we collected, the epithelial cell always cluster with the non-neuron a little bit better. But for the human data, unfortunately, we don't identify much cell populations for epithelial cells. Yeah.
We're gonna interrupt you a lot. but
Yeah, of
we
course.
go long, so don't worry about it too
Yeah.
much. So because there's such a gradient in metabolic activity in cortex, you have this striatum, which is generally, if you go to globus pallidus, it's a lazy part of the brain. If you're in cortex, it's pretty busy. If you're in the suprachiasmatic nucleus, it's super busy. I'm just wondering whether this is intrinsic or it's just, hey, I'm in a neighborhood. I've got all these changes. I've got a whole bunch of glutamate flux. And by God, I have to open up this piece of chromosome. So I guess the question is, are they fundamentally different, or it's just, if you took a cell type and plunked it in an alien environment, would it quickly convert to another type?
Yeah, I think that's a good thought. Actually, I think if we sample the cell in different time point, we can better address that, or we can do this crazy experiment to put it in somewhere else. But at this moment, it's hard to say. This is just what we saw. I think one way we try to interpret that, because we know the brain tumor coming from different region probably have some issue, but I agree with you. It probably also related to the environment and where the cells are located.
So how many markers are you using to define this cell type? For example, you using the CR3, CR1.
Yeah.
If you have multiple markers to use to define cell type, do you think that that's because the difference between different regions is attributed to the different markers instead of the cell type itself?
Yes, that's a good question. So there is a long debate about what is the cell type in the brain initiative. So for many people working on single cell RNA, they can set a cutoff, say, when you do differential analysis, if you can find 3 or 5 differential genes, I call this a novel cell type. Here in our study, I did not do it in this way. I do it, the cell mutation, in a more conservative way. So for the major cell population I listed here, I list 3 to 5 well-known marker genes for the major cell population. And then we also confirm whether the cell population we annotated for certain cell type coming from the right brain regions. And then in this level, we integrated with the RNA generated by an institute to confirm the annotations. Yeah, I would say this is probably not giving you thousands of cell types, but we are pretty sure this 100 cell types are very robust. They're the 2 major cell populations. Okay. So, well, thank you for those questions. I guess now I hope I convince you. This is just the way to analyzing the data at the beginning, right? I mean, the analysis of that, the main purpose is to find where are those areas. Those areas could be cell type specific, could be regional specific. And then, like I said, well, many of those risk variants actually located in those areas. And then from the GWAS analysis and fund mapping, people already try to prioritize the risk variance. But people are still don't know which is the disease relevant cell types. I think that's in my opinion, this is one of the most important application for single cell data set. So the next point we're asking is whether the disease risk variants are enriched in the enhancers for certain disease. So this is the LDSC analysis where you can consider this as a regression analysis. I show you a heat map. Every row is a GWAS study. The risk variants are identified for different neuropsychiatric disorders. For every column is the cell type we identified from our data. So we basically ask whether the risk variants identified, for example, for schizophrenia, can be explained by the open chromatin region or the peak regions we identified for certain cell type. So
I love that, the schizophrenia and intelligence are right next to each other.
yeah. It's also a very surprise to us. Well, in this way, I think I actually want to emphasize this figure. I think sometimes for the single cell data, it's not about annotate new populations, annotate more cell types, right? It is about associate the risk variance to the most disease relevant cell populations. I can show you several examples. For example, the schizophrenia, they are associated with many glutamatergic neuron cortical region as well as GABAergic neurons. I think this is known. I think for many nature genetic studies also find that. Alzheimer's disease is quite enriched for microglia. This is also reported by many people. Similarly, you can see like major depression disorder associated with certain populations of the intertalacephalon neurons in the cortical region. And I can show you more examples of that.
But Alzheimer's disease is a disease of neurons, not a glia.
Well, many years ago, Inco and Manolis Callas also thinking like the AD is associated with neuron. But to our surprise, yes, it is. But if you study the genetic variations in this level of regulation, it is enriched in the microglia. I think one disease, it is complicated. We do a lot of EQTL, CAQTL, but that is only focused on the gene regulation level. The cell-cell communication also matters, and then environmental factor also matters. Eventually, you can develop that disease. I think here, one assay cannot address everything. I think here, the TAXIC data just try to explain some of the variants in gene regulation level.
It's going to help a lot of us. Most people here aren't neuroscientists. So you've got all of the layers of cortex over there, mainly glutamatergic, and a whole bunch of interneurons. And then what is the purple for Alzheimer's? What's MGC?
Oh, that's the microglia. Microglia.
And the last ones there, SN.
Smooth muscle cells. Yeah, we capture a little bit of that.
Thank you.
Yeah, a lot of the AD associated risk gene are enriched in the microglia, including TREM2, CD33, a lot, MSC4A.
Okay.
And I was wondering, when you take this risk gene, or if you look at the raw side, you cover the multiple candidate gene, and are you only look at the risk gene with the leading step, or how do you define this risk gene?
We're now using the fermenting results. We're using the summary statistic from the GWAS study. And then we're now focused on the risk genes. We're just focused on the open chromatin regions we identified. We included all.
No matter whether you use meta-analysis or not, you still have to identify the raw side, right? You identify the raw side, then you still need to select the candidate or the risk gene. How do you define that that's your risk gene. How far from your chromatin peak to the lower side? And do you have any threshold cutoff, or how close do you think they are close enough?
Oh, for here, we just overlap any significant variants identified from GWAS study with the open chromatin regions. They must be inside the open chromatin regions. So we took that into
So you're
consideration.
already using the fund mapping result.
No, we're not using fund mapping results. We're using all the significant variants identified from GWAS study. And then this is our first step. So the purpose is we want to include as many as significant variants as possible to pinpoint disease relevant cell types. And then starting from there, we can do fine mapping to further identify the most causal ones.
One question. So I remember you said that the tissue for your study is from 3 normal donors. So how do you went from normal people to this disease?
Oh, the normal people can still have the open chromatin region open, right? They are functional as an enhancer silencers, right? It's just those regions probably is closed or even more exposed in a disease condition. It doesn't mean they're not existing.
But the two, there is no established genetic traits.
There is in the GWAS.
There's some not well established. There's some suggestions, but it's not hard evidence.
I think GWAS simply tell you within.
It's hard.
Yeah, it is a hard question. I think GWAS simply tell you for certain disease, right, within this cohort, whether you see a higher chance to identify this risk variance. And then we're trying to explain that by open chromatin regions. I think I can answer your question in another way. I think it's better if we have the profiles from disease conditions. Yeah, but
Either way.
but
It seems like in some diseases you may degrade the signals that you're using to classify. Do you see that? Does it get harder to classify cell types accurately in some disease conditions?
I think cluster disease conditions is actually how to correct the genetic background. I think that's the main issue actually. For those 3 individuals we collected, we don't see much like donor effect. But I do know some of my colleagues, they are collecting disease conditions. Because the genetic background is so different, you will need to do certain corrections to align the similar populations. Yeah, I think, yeah.
So you take 2 factors into account for diseases, like first one is the chromatin openness and the interaction between the promoter and enhancer regions. And the second one is the somatic variants. For diseases, how can you evaluate the importance of certain factor? Like, do you stress out this chromatin openness is more important in these diseases, or do you think the GWAS is more critical for this type of diseases, or how do you measure the importance of 2 factors?
I think the GWAS analysis give you the significant risk variance, right? But say you have 10,000 of those variants. How to annotate the function of those variants is the question we're facing. For some of them, you can annotate it to the coding region. Probably there's a missense mutation, nonsense mutation. So that's the way you explain the function of those significant risk variants. But like I mentioned earlier, like 90% of those significant variants are not really located at the coding region. I think that is why the open chromatin region identified become helpful. We try to further annotate the potential functions into, for example, you said the enhancers or silencers. That's the goal of this analysis. Okay, I'll keep going. So I guess this is the first step, right? The second step is we really want to figure out what is the potential target genes for those CREs, and then how to link really the risk variants to a gene we care. So to do that, actually, we borrowed the RNA-seq data from Stan Linderson's group. You can measure the gene expression levels from RNA, and then you can identify those CREs or switches from the tag-seq data, right? And then I guess our task is really to draw a line between the switches and the light, or between the gene and the open chromatin regions we identified. So this is a two-step. The first step, we just try to calculate the covariance of two open chromatin regions to see how likely those two genomic regions are open at the same time across different cells. And then the second time, because we do the integration with the RNA-seq, we know the gene expression for a certain gene. So we can further calculate the correlation between CCRE accessibility and the gene expression levels. So in this way, you can feed a known distribution and then define a cutoff to further figure out the potential target genes for the enhancers. So this is the number. So I told you I identified 500,000 of those CCREs, but eventually we're only able to annotate 100K-ish putative enhancers from this data. And then those putative enhancers have been linked to certain K putative target genes.
How far you go from the gene to identify the CRE, 1 kb, 1 million, or another chromosome?
Yeah, the average distance is 171 kb region. You definitely have the CREs located pretty close to the promoter region, but you have long distance enhancers.
So the disease GWAS are saying that overlap the open chromatin regions, have you checked to see if they're much with EQTLs?
No, no, we haven't been able to check this. Yeah, so this is just showing you two heat maps to confirm the prediction we have. So left side, you'll see the CREs we identified. They're quite cell type specific, and then you see the corresponding putative target genes are also highly expressed in the certain cell type. Okay, so why this is useful? Why we need to do this prediction? Because for from the GWAS study, we can identify the risk variants associated with schizophrenia, right? But you didn't know what's the function of that. You try to annotate that. So the open chromatin signal we captured indicate here is the potential enhancers close to the NAB genes. NAB genes is reported associated with depression and schizophrenia. Probably from EQTL studies, you can potentially link this gene, this risk variant to this gene. And then the tag-seq data just help you further annotate the function. This risk variant probably disrupt the function of this enhancer so that they have a function on the NAB gene expression. So this is an example for neuron. I have another example for non-neurons. This is an example from microglia. You can see there's also actually this is the causal variance from the fat mapping. This risk variance is located as a microglia enhancers we annotated. You can see this enhancer is exclusively open in microglia, but not in the other cell types. And then from the gene expression and open chromatin covariance analysis, we can link this enhancer to the gene. So the hypothesis is probably this enhancer have a function to regulate the gene expression of B1. B1 is have been identified as a risk gene for AD.
But in this case, you think it's the model next door, or you think it's a distal CRE?
Yes,
Okay.
yes. So actually,
That gene looks pretty interesting. Well, why don't you like the XR gene?
uh,
How do you know they didn't get it wrong?
this is probably an annotation of the pseudo gene. I just load the annotations. It doesn't mean there's a transcripts of this gene in the microclip. Yeah, yes, there are none. So actually, CRISPR-Cas tested this enhancer. They tried to use CRISPR to knock out this enhancer, and then they do see the gene expression of B1 decrease in the microclip.
And an interesting comparison with the FTO story, being IRX3, where the FTO is one of the most famous genes associated with obesity, and GWAS is huge, it's like lots of 20. It turned out that it's just that happens to be a variant and an enhancer in the intron of FTO that actually controls IRX3, which is like 250 kb away.
Oh, yeah, could be. Yeah, could be.
I'm just thinking, but I wouldn't trust the annotation for BIN1 unless there's killer data. Probably there's killer data. It's a well-known ADG, but there was killer data for FTO also.
I see. So keep going. So I guess so far we cannot find a way to find where those CREs try to link it to potential target genes, and then the last thing we really want to understand what is really the function, how those variants cause the gene dysregulation. So to do that, actually, we can do eQTL this analysis. It's just recently the machine learning approach or deep learning approach become very helpful to help you prioritize the function. So I was actually inspired by this model called Informer, developed by Google and DeepMind. They are trying to predict the gene expressions from the DNA sequence. During the pandemic, we got a lot of help from UCSD's CS department, especially for the GPUs. So I trained a model. I won't explain the details, but the input is the DNA sequence. And then I try to predict the attack signals, the different cell types. So the idea is, yeah, as I said, the input is the DNA sequence. We try to predict the potential functions. In this case, the potential function, it is the open signals we captured. We adopted architecture called a transformer, multi-hat transformer. Actually, this is the core algorithm behind the ChatGPT. So they are training transformer-based method. I can explain it even further. For example, if you try to translate English to French, right? It's actually very, very easy to find the words with similar meaning. You simply look at a dictionary, right? When you're programming, you just check out a lookup table, you can find that. But the tricky part is, if you want to have a sentence, have a meaningful, if you want to generate a meaningful sentence, you have to put the words in the right position. They probably not follow the exactly same order, or their distance are different, right? So the transformer architecture can better capture the relative distance between meaningful words. That's why they're very useful or very popular for natural language processing. So if you think about the DNA sequence, the combination of nucleotide, they can form the binding site of the TF. If you consider that is some of the grandma is using in DNA, you can count on this architecture to figure out what is the combination of nucleotides give rise to a certain functions. That's the idea.
How to compare with the CNN?
We actually have a CNN layers there.
You have CNN.
CNN layers, you try to virtualize the motif informations. because we're trying to predict the signals in 100 kb region, that's a long distance, CNN will lose attention to long distance. Transformer have better capture the relationship between long distance.
Then how many head do you use?
For this one, we use 8 head.
I just want to know that this DNA sequence is like linear, ATCG, or it's like a matrix when you give input to the transformer.
Yeah, this is a matrix. We generate this in one hot matrix. Here is each row is A, C, G, T. And then if the first position is A, I will code it at 1, 0, 0, 0, right? And then the next position is C, you will code it as 0, 1, 0, 0. I don't want to say like the current, any of the deep learning model is perfect, but they do learn how to predict the signals. I hope you can appreciate, well, they can pretty well predict some of the cell type specific signals, as I indicated here. And this is actually the examples I showed you earlier. This is the microglia specific enhancer. And then the model can pretty well predict the signals here. Well, you may ask why we treat this model, because we already generate the data. We know where are those CREs located. We know the signals. But the reason is you want to let the model to learn from the sequence, what combination of sequence will give rise to a function, right? And then the key is really how to use this deep learning model. You can insert many of the mutations you care about in silico. It's easier to do so comparing you design the sequence to CRISPR, knock-in or knockout, right? And then you can count on the deep learning model to give you a prediction. For some of the risk variants, probably the model will predict, well, you probably have a lower open prompting signal. Some of the variants doesn't matter. Whatever nucleotide you change, don't have a change of the function. For some of them, you probably see an increase of the function. So you can really use this kind of model to prioritize some of the functions. So this is what I did. I did the histological mutagenesis for this enhancer. So you can tell in this little heat map, some of the color is in dark red, some of the color in dark blue. That basically means if you mutate that single nucleotide, if the model predict you have a higher level of open protein, you will see a dark red. If you see, like, if I mutated this nucleotide, the model predict will lose the function in a dark blue. So first of all, within this enhancer, not every nucleotide matters. There are certain regions the model predict, if you mutate that, you will see a loss of the function. And then it makes sense. For example, for those regions, the model predict have a higher effect of the function, they actually overlap with some of the transcription factor binding site. What I'm trying to say is, at least this is the example to show you the model do learn something, you learn the meaningful informations instead of randomly like capture the informations.
But this model is not trained for the brain. For example, for the liver.
No, this model is.
It takes the liver peaks in the peaks in the liver cells as well. It's not trained for the brain. It's a general prediction model. Am I right?
Yeah, it's not a generated model. I only train this on brain data, but you can train it on liver data or whatever cell types.
If you use a 10 kb, right, it contains multiple peaks, right, multiple CR.
Yeah.
And, do you also use the position embedding there, or?
Yes, I actually train on 100 kb. It's not 10 kb.
100 kb.
Yes.
Then there's a lot of peaks.
Yes, for transformer architecture, you have to incorporate the positioning embedding into the model. So we're focused on this region because this region have the causal variance in AD. And then this model actually predict, well, if you have this mutation, you're likely to see an increase of the function from C to T. Why is that? This is to our surprise. So if you take a close look at the sequence, in the reference genome, this is TAAACA. But in the patients, in the AD patient, you will have a mutation from C to T. And then this sequence will change to TAAATA. And then they actually created another potential binding site for TF from the M2 families. We further confirm whether this in one gene, this two TF is expressed in the microglia by using the data generated by Allen Institute. So what I'm trying to say, this is the approach we can counting on to further prioritize the potential functions, so that you can have a list of risk variants, say top 10 or top 100. You can design the follow validations. So this is my first part. Overtime, but I still want to finish this. So
His fault.
no, this is a good question. Well, we generated this map. We identify some of the candidate cis-regulation elements, and then counting on those open protein regions, we can explain some of the risk variants for neuropsychiatric disorders. I really want to emphasize, I think the most important application of single cell data, it is to associate disease to disease relevant cell populations. I think that's the material I want to talk about. So this data have been published, and then there's many other modality data published into this package. And then I was also very fortunate getting involved in the mouse data analysis. We generate the similar things for the whole mouse brain. And then all of this data actually is available in a web portal I built a couple of years ago called Cathless. I really like this name, and then my wife designed this logo, so thanks to her, I have a nice logo here. So you are not only able to explore the human brain data, but also many other data set. Another good news I want to share is my lab member is actually working on updating this web portal. We're collecting more epigenetic data for different assays. We try to integrate them together. In the future, I guess we're offering options for searching the genes for variants you care about, browse the data, and then we try to train this model for more cell types so that you can make predictions. Hopefully this will be, the second version will be online like this year. Yeah. It's a tons of work, like aligning this. Okay, so I will be quick. I don't want to use too many of the times. I want to quickly talk about now we have the reference map, right? How to use it, how to use those reference maps. In our opinion, there's an active, there's a lot of studies actually interested in profiling different disease conditions from different individuals. And then there have different animal models explore different disease conditions. And then it's very interesting to actually compare across species. I will touch it a little bit. So first of all, I think using those data can help you prioritize the risk variants in disease relevant cell types. This is a great example. So people identify one single mutation in IDH1 mutated glioma. So they know this risk variance is located in a human, in a brain specific insert. They validated that in the engineered mice embryo. And then you see this inhibitor is actually active in many of those brain regions, including the spinal cord. So they are not able to identify what is the cell population related to that, but they are able to test why this risk variance matters. So in the normal genome, you have an A here, but in the patient with glioma, they mutated to G. Actually, this mutation disrupts a binding site of 2 or 4. So as a result, you will see the higher expression level of MYC gene in more than 40% of the patients. So like I said, this is great, but you still want to find out what is the most relevant cell types, right? I can give you a quick example. This is the, at least to my knowledge, this is the latest GWAS data for brain tumor. And then they do identify many new locus. And then even right now, we are still trying to generating data from the brain tumor sample. But you can counting on the data we already generated. You can see there some of the new locus, it is located in the astrocyte specific cancer. So probably astrocyte, it is the cell population or astrocyte-like cells you should be considered for further validation. Okay. So the second step, I know there's a very nice audience focused on addiction and associated behaviors. Actually quite interested in that. So I did a little bit analysis. So this is so far all the risk variance associated with opioid disorders. I can collect it from GWAS catalog. And then I do the enrichment analysis, try to prioritize what is the potential cell type are involved in this. I think this result makes sense, right? It is associated with MSN. So I got a lot of help from the core from Scripps Institute, generate this IVSA model, and then we generating the single cell T-seq data and RNA-seq data from the nucleoside. I want to point out, actually, once we generate all of this reference map, you can directly use it instead of generating that, because that's the normal condition. So long story short, the cluster makes sense. We identify the most major cell types, and then the treatment control very well, which we have. The real issue is there's not so many significant differential genes or peaks we can identify from this data. It worries me a lot at the beginning because with this little differential genes, it's not even possible for a significant downstream analysis or the goal term or enrichment analysis. So later I think about it in another way. They probably not there's significant change of the gene expression. Maybe there's change in the expression variations instead of suddenly change the levels. So how to visualize that? Actually, I adopted an approach from this Nature Biotech paper. They developed a new way to show the data in a 2D dimensional space. So in a normal t-SNE embedding or UMAP embedding, you only care about the neighbors. So you can calculate the similarity between every cell, so you can link them together. But actually, when you calculate the similarity between any two cells, you can also measure the distance between two cells, right? If these two cells are very much similar, the gene expression are very much similar, they're close to each other. If there show some variations, they're further to each other. So this tool just also included the local radius or the distance between two cells into consideration. And then they're trying to visualize it in a different way. So in this case, for those cells that are more sparsely or more loose distributed, that means between any two cells in here, they show a higher level of gene expression variations. Does that make sense? Okay. So if you take a look at the data, you visualizing in the normal way, using UMAP or the dense map, you do see some of the populations are starting to have a loose distribution in this normal values. Right? If you compare, if you compare here is the control. I'm sorry, this is first one. So this is the control. This is the treatment. So you see under treatment, this population is more loose distributed. That is treatment. We're giving heroin treatment. Heroin. Yeah. So instead of in the control, those cell population, their expression level are tightly controlled. So this is actually inspiring me to do further analysis. I look at several genes. Again, I was not an expert in this field. So you see, the normal approach, we call a differential gene, we care about the mean values a lot. Of course, we care about the variations between individuals, but we care about the mean values a lot. But actually, the mean value doesn't change too much. Instead, the range of the gene expression change a lot. I think that totally makes sense. Here, the violin plot shows the expression level for this gene across all the cells, either from control or from carrying treatment. I think that basically makes sense. If you give a treatment, you're not expecting all the cells suddenly change, right? They're suddenly change all the expressions for a certain gene instead of a subpopulation of the cells changing their expression levels.
Did you see this several times or just in a one mice experiment?
I'm sorry.
Did you see in a couple of mice this kind of distribution?
No, we only saw this in 2 replicates. We don't have any time point or this is a acute injection. That's the only data we have at this point. Yeah, in similar way, you can identify more genes. Actually, they show a higher range of the gene expression variations. So similarly, you can do the similar analysis for ataxic data, right? Because the nucleosome position could be changed. You can measure whether this reads is close to the potential TF binding site or further close to the TF binding site. In this way, actually, for many of those peaks, we do not see a difference if you only care about the race counts. But if you're using this probe, you can see the variations between peak. For example, you see there's a little dip in this peak that probably indicate a potential binding of the TF or a TF footprint there. Okay, so this is the second example I want to show. I want to point out to many of those normal condition reference maps can be directly used to compare with many disease conditions. And then the third thing I want to point out is actually with the multi-omic data set, you can further dissect the gene regulation programs. So if you can integrate all of these informations together, it would be very helpful. For example, I show you one case. This is the data I collected from many papers. They're from the cortex. And then first of all, you can very well align them together. And then I show you one example. The CUX1 is the marker gene for telocephalon neuron from layer 2-3. You definitely can see the high expression of the RNA, not surprised. From the ataxic data, you just identify many peaks there. And then you do need some additional information. For example, the histone modifications we can capture from paratag to further prioritize what is the potential active enhancers. Because there are so many peaks there, you want to narrow down to an enhancer for downstream analysis. Of course, now the spatial transcriptome is very popular. This is a population is very easy to find, but you can count on it to look at where the spatial location of that. And then if you're familiar with the Hi-C data, the Hi-C data also indicate, well, this is the enhancer potentially regulate the gene expression of the CUX1. The last part I want to talk about, it is the conversation between human and animal models, which is very much interesting to me. So we're counting on animal models for many human disease research. But given the difference in size and many other morphologies between human and mouse, I'm just curious, what is the conservation of the divergence between human and mouse? Right? Well, the first step is I try to align the similar cell populations together. So this is a pseudo-bot level analysis. You just extract 20-ish cell types from the human cortex and 20 cell type from the mouse cortex. If you only counting on the gene expression levels at the ortholog genes, without any correction, you probably cannot really align them well. This is also true if you look at the INCO paper. They also say the similar species are probably close to each other instead of the similar organ with similar tissue. So later I think about, well, I also have the toxic data, maybe I can look at the CREs instead of the genes. Same thing, there's a lot of turnovers of the CREs in human and mice. You are not able to well align them together. But there's a hope. So the TF motifs are very much conserved. This is known from many studies. They said for most of the TF, their binding site are very converse across 2 billion years of evolution. But humans do have some of the TF are quite unique. So we counting on the TF sequence, using that as a feature, do not do any correction. You can pretty well align the similar cell populations between human and mouse. So although there's a lot of turnovers in gene expression and CREs, but they all follow the similar rule in gene regulation. So we later push it to single cell level. Now you can very well align the similar cell types between species. So once you align the similar cell types between species, the next thing we want to ask is whether we can identify the autolog or conserved CREs between two species within the similar cell types, right? We basically cluster them into 3 major groups. For sure, you will have some of the CREs, no matter how hard you try, you just cannot find the similar sequence in the mouse genome. And then we call them human-specific elements. Some of them is very easy to find. Most promoters, they are very well conserved between species. We call it open chromatin accessibility conserved ones. You'll also see many similar sequences between human and mouse genome, but in either species, they're not using it. They're not using this sequence as a regulatory elements. So this is the general distribution. We find actually, if you only compare human and mouse, I know the evolution distance is quite far between human and mouse. You can introduce primates, but the comparison between human and mouse, you'll see 40% of those elements are human specific, and then 30% of them are human mouse conserved. And then there's another 27 of them are divergent. They have similar sequence, but they're not functional in a similar way. This is not biased to certain cell population. This is quite similar or consistent across different cell types. Okay, I'll show you one example. So I'm very much interested in one subpopulation of the VIP positive coverage neuron. You see there's a subpopulation of cells have this gene, CHRNE2, have a higher level of chromatin accessibilities. So my friend Antonio helped me to validate do exist this kind of cell population. You do see the expression of VIP as well as the CHRNE2 in the same cell using RNA-scope approach. Why this population is interesting, because later I find, well, this is a little heat map of gene expression. You see this CHRNE2 gene, it is expressed in some of the subpopulations of VIP positive neurons in human cortex. But if you check the mouse data, in mouse, you also identify many subtypes of VIP positive covered neuron, but this gene is not expressed at all. This also. If you further look at the open chromatin signal, you see there's a human-specific enhancer. You're not able to lift over or find the similar sequence in the mouse genome. Yeah, and then this is predicted to regulate this cholinergic receptor, also supported by IC data. And then still very interested in human-specific ones. We find actually human-specific enhancers are quite enriched for the transposable elements. Like I mentioned earlier, the transposable elements have some evidence to show they're contributing to the brain development. So I try to identify the cell type specific transposable elements, and then you see some of them are quite enriched in microglia. And then there's one called LTR13A. This is actually known, have been validated these transferable elements can act as an enhancer in leukemia, but I just don't know whether they function in a similar way in brain. But that's a good sign to find some data to supporting that. And since we have the data from different cortical region, we can plot the aggregated signal at this LTR13A across different brain regions. So what you can see here is those are the cortical regions. You see a higher signal of open promoting at this transposal elements. Here you will see some of the lines in yellow or greenish color. Those are the signal we capture from the cerebellum or from PAMS. So at least they're showing maybe this transposal elements is more active or more in the open promoting state in the cortical regions. I think this makes sense because if you compare human and mouse, right, the major difference it is the cortex. We have more complicated functions. And I don't really want to make a conclusion here, but we do have the data from different individuals, they're in different age. We do see there's a little trend associated with the aging or different ages. But again, this is the data set is too small. We cannot make the conclusion. Well, to further confirm whether this is likely the active transposal elements, if you check the RNA-seq data, single-cell RNA-seq data, you do see there's a potential transcriptions for that. Okay, this is the last thing. I hope you still remember this, the LDSC analysis where I try to associate the risk variance to cell populations. Now you can do this for the three major group of CREs separately, right? You can do it for conserved CREs, you can do it for divergent CREs, or you can do it for human-specific CREs. I think the good news is you still see the strongest enrichment for human mouse conserved CREs, but there are tons of risk variance enriched for human mouse divergent elements. I want to point out the Alzheimer's disease. You see the Alzheimer's disease, it is an outlier. You see the strongest enrichment in human-specific CREs instead of in human mouse conserved CREs. I know the Alzheimer's disease is complicated. There are probably many cell types, many pathways get involved. At least in this level of the gene regulation, it is true. There's many human-specific mutations or patterns are associated with that. You cannot really find it in the mice. That probably indicate, well, if you study genetic variants, probably iPSC-derived microglia line is a better model comparing to a mice model. But if you study cell-cell interaction or like how they respond to the tau protein, well, mouse is still a good model for that, right? That's interesting. So again, with this reference map, you can really try to prioritize the risk variance in disease-relevant cell types. You can you can use this reference map, especially from the normal condition, to compare with certain conditions. And not only you can capture RNA and open protein, you can use different information to further dissect the gene regulation program. And then lastly, I just fascinated about human and animal models comparison. That's it. A lot of people got involved in my previous lab as well as my current labs. Thank you so much.
You must go to the airport.
I have
Yeah.
Question on the draft. Yeah, thank you, everybody. Can you maybe talk to him on the way out?
Yeah.